Hello, I'm going to explain how the cell membrane works and how molecules diffuse across the membrane and how water moves across the membrane. And then I'm going to go over the diffusion lab, diffusion and osmosis lab. So first, let's take a look at this slideshow, and it shows the membrane, and it shows that different things can come in and out of the membrane, and other things cannot go in or out. So that's what we call selectively permeable. It's doesn't let just anything come through, and so that's selective. And permeable means to be able to go through. So some molecules come in, others are out. This is a kind of a model of the membrane. You can see that it's actually a row of lipids here and a row of lipids here. And that kind of double lipid layer allows it to kind of be flexible and moving, and it also is what allows things to kind of pass through or things to not pass through. That kind of water-loving and water-hating sides of the lipid molecule. So the membrane, it protects cells, it controls what goes in and out, it maintains ion concentrations of various things inside and outside the cell. Remember, everything has to be balanced for the cell to function correctly, and this membrane is what we call selectively permeable. Okay, so an example of how our membranes work is the blood vessels and nerve cells in our brain. So the blood-brain barrier, and some of the molecules that can pass through into brain cells are water, carbon dioxide, glucose, oxygen, amino acids, alcohol, antihistamines, different viruses, like HIV and meningitis can also cross over. And that's really important in knowing how we respond to different chemicals, and also how we get certain things that our cells need. Okay, so there's two ways that, well, there's more than two, but two ways that we're going to talk about that molecules move. So the first term that you need to know is diffusion, and that's just things moving from a higher concentration to a lower concentration. So just everything evening out. From, if it's really concentrated in here, then the molecules are gonna just keep moving until they're evenly spread out. And that happens like if you spray perfume in a room, and then soon it just kind of, the molecules keep moving until they're evenly spread out. Here you have an example of a membrane, and this membrane looks like it allows this green molecule to get through. So the green molecule is going to keep moving through until it's even, and that's called diffusion. So it doesn't require any energy, and it just kind of easily passes through. And a lot of molecules will pass through our cell walls through diffusion, or cell membranes. Here you have, high concentration of the purple molecule, high concentration of the green on this side, and the green will then move to a lower concentration, and the purple will move to the other side until everything's even on both sides. So this is called diffusion, just moving from high concentration, where there's a lot of whatever molecules you're talking about, and then kind of evening out. This concentrated area is going to go to where it's less concentrated. Okay. Now we're going to look at osmosis, which is just the same thing. It's diffusion, but it's diffusion of water. So instead of talking about these little things floating in the water diffusing, now we're just going to look at how does water move across a membrane. And the way water moves across the membrane is the same way, except this time we're just talking about molecules of water. So it's the diffusion of water from an area of high water concentration, so that where there's a lot of water. So here there's all these water molecules in here, and there's fewer water molecules here. So the water is going to go from where there's lots of water molecules to where there's less water molecules. And so high concentration of water to a low concentration of water. These green things, apparently they're too big to move through the cell membrane. The purple things can't move, but water can move easily through the membrane. Okay, and then here's another example. Here's a membrane, and it looks like the purple things cannot cross. If the purple things could cross, then they would also diffuse until it's even. But water is going to go, this water has a higher concentration of water. Another way to look at it, it's more pure. So it's more pure water molecules. And here it's like less water molecules because it's mixed in with all of these purple things. So water is going to go from high water concentration to low water concentration. More pure to less pure. So you can see that happened here where the water crossed the membrane and now there's more water on this side. And that's, this is osmosis. It's just the diffusion of water. And I remember when I was in high school, we used to say, I wish we could just lay down on our books and have all the information go into our heads through osmosis. That sounds super nerdy, but it's actually inaccurate because the information from our book is not water. So using the word osmosis was incorrect. We might have said diffusion of the material. That might have been a little more accurate, but still kind of cringy. Okay, so those are the two terms you're going to need to know for your osmosis and diffusion lab. Diffusion is just movement of any molecules, and osmosis is the movement of water molecules going from high concentration to low until it's equal. And even after it's equal, the molecules keep moving, but they just are equal. Okay, a couple other terms you're going to need to know is solute, solvent, and solutions. You don't have to memorize it, but basically a solution is a solute and a solvent. A solvent is the liquid that you are adding the solute to. So solute could be salt, it could be sugar, it could be any sort of chemical you're adding to the liquid. And it's dissolved in the solvent. So when you take a solute, dissolve it in the solvent, then you get a solution. Okay. For the egg lab, you are going to be looking at a couple solutions. So you're putting an egg in either salt water or fresh water. Actually, not salt water, fresh water or sugar water, which is the corn syrup. Okay, so if you put something in fresh water that hardly has any solutes in it, okay, so here's like our pretend cell, and there's lots of different solutes in a cell. There's lots of things floating around in the cell. It's not just water, but if you put it in pure water where there's barely anything floating around, the solutes in the solution is going to be less than the solutes in the cell. There's hardly anything in here, but there's more solutes in the cell. So that's called a hypotonic solution. Hypo means under, so less, like a hypodermic needle or a hypothermia. Those are all like under the dermis, under the skin, under below the normal temperature, hypo. So here the solutes in the solution are low. They're under what the cell should be. Iso means the same, so you can see the solutes are the same. The number of solutes here equals the number of solutes here, more or less. Okay, so the water concentration in both is about even. Okay, here you can see the water concentration is higher because there's more water molecules here than here. Okay, hypertonic. Hyper means above, so there's going to be more solutes here in this solution. Hypertonic means the solutes in the solution is greater than the solutes in the cell. So you can see that this is hyper, and this will be like our egg that's going to go into the sugar water or the salt water. Okay, so this is going to be our egg that's in the fresh water, and this is going to be our egg that's in the sugar water. Okay, these are some more examples of what we just said, and this is kind of looking ahead. What do you think is going to happen? Because the water is going to go from more pure to less pure. It's going to go from more water to less water. So it's kind of predicting. Okay, so here it's more water to less water. So the water is going to move from here to here into the cell. That's our prediction. Whereas here, the water is more concentrated in the cell. There's less water concentration outside of the cell. So here it's more pure, here is less pure water, and it's going to go out. If you're looking at the green things, that's not what we're talking about for the egg lab. The green things is just diffusion of these solutes, but they can't go through. The green things can't move. It's only the water that's going through, which is why we're going to call this osmosis, because it's the diffusion of water and not these other molecules that can't fit through. Okay, so here's some examples of what's going to happen in the hypotonic. Sometimes when you put like vegetables in water and you keep them in the refrigerator, it kind of causes water to go into them and it stays nice and fresh and crisp. Carrots, celery, sometimes you see them at the supermarket doing that, and that's because they know that the water is going to move into the cell because it's going from high concentration to low concentration of water, and that keeps the cell wall keeps it from getting too much overload, but it also keeps it nice and crisp, the vegetables. Isotonic, no, everything's equal, so you're not going to have like major drama. Hypertonic is when you're putting a cell in a solution that has a lot more solutes. So the water is going to go from inside the cell to out of the cell. And so the water is going to come out and the cell is going to shrink. So this is a cell in salt water. If you put it in salt water, then there's lots of salt here and there's not a lot of salt here. The salt can't move, so the water is going to move. The water is going to go from high concentration of water to less concentration of water. And that's what happens when you put salad dressing on your vegetables too, where all the water starts moving out, and if you leave it long enough, everything gets wilty. Okay, so that is what you need to know for the lab ahead of time. Here is one of the solutions you're going to use for the diffusion part is iodine. Iodine normally is this color. It's called amber or rust color. If you drop it on your skin, it just stays this color. Sometimes you see it in hospitals where they like clean the skin, antiseptic, things like that. But if you drop it on something that has starch, it turns blue-black. Okay, bluish black. So here's a potato. Potatoes are very starchy, and so that turned black. If you put it on your clothes, it'll turn your clothes black because normally your clothes are from cotton, which is a plant, which has a lot of starch, cellulose in it. Okay, so here you dropped the iodine in water. No change, it's the regular, there's no starch. Obviously, if you put on starch, it's going to turn blue-black. Milk does not have a lot of starch. It has a lot of sugar, but it doesn't have a lot of starch, so it just stayed. It looks like it got a little black down there. Tofu is mostly protein, so that did not turn blue-black. And the apple looks like it has a little bit of black. Apples have a little bit of starch. I wonder if milk has starch. That looks pretty black to me. Okay, so let's take a look at our lab. I was going to make a video, and maybe I will, but someone did a nice video of the lab, so I'm going to let you watch that. And you're just going to go ahead and answer these pre-lab questions, which are coming straight from this introduction. Okay? And then it tells you how to do the lab, which you'll just watch her do. But basically, you are going to see her put a little cup of iodine and water. So that's iodine and water. And then she's going to make a little, bag and fill it with starch. And this bag is representing our cell with a selectively permeable membrane. So that either it may or may not let the starch in or out, and it may or may not let the iodine in or out. Okay, so this was the setup. And so what you need to do is take a look here and go ahead and put what is the initial color of the solution in the beaker and what is the color of the solution in the cell. Okay. And then you're going to wait 15 minutes and see what happens. Dun, dun, dun. Okay, so here you can see, this is the before and this is the after. You can see that the starch turned black. Okay. So now you have to think, why did that starch turn black? What happened? What happened? Okay, so you're going to go ahead and put the final color of each, and you'll notice that the iodine in the cup still stayed amber, okay? But just the starch turned black. And remember, in order to turn black, you have to have starch and iodine touch each other. They have to come in contact with each other. Okay, so you're going to go ahead and answer these questions, and then you're going to just draw what happened, what moved across the membrane. So you can put, we started off with starch in the middle, so you can put a little S here and iodine here on the outside. And I want you to draw an arrow showing, did the iodine move into the cell or did the starch move out of the cell? Okay, so draw that arrow for which one happened. Okay, and then you're going to answer that question, and then you're going to do your final question is, what is diffusion? Okay, and if you forgot, you can go back to the slideshow. All right, the second example of diffusion, we're going to look at osmosis, the diffusion of water. And again, unfortunately, we can't do this in class, so we have this great video of someone who did this, okay? And I love this because she kind of like talks a little bit about what happens if you drink salt water and all that. But she goes over, hypotonic, hypertonic again. She tries to give her dog salt water. Okay, that was a little scary. Okay, there was a stunt, she said. Okay, and then she talks about how the membrane of an egg is semi-permeable. So when you peel off the egg and then there's like that membrane right under it, it's kind of like a papery thin cover. That is the cell membrane of the egg. And so what she does is she has to get the shell part off and just leave the membrane. And the way she does that is by putting vinegar on it, and that will dissolve the shell and leave 2 lovely eggs. Okay, so she did that for 3 days. And then she's going to go ahead and weigh each one. So that will be your initial mass. And then she's going to put them in different solutions. Okay, I might have to add hypotonic. Okay, so 1 egg she's going to put in a hypotonic solution water. Okay, so that means there's less solutes in the water than there are in the cell. There's more. This is more pure water, and this is less pure water. Okay, and here's hypertonic. This is a corn syrup. So there's lots of sugar in here. So that means there's more solutes in the liquid, and then there's less than the solutes in the cell. And so we're going to think about what is the water going to do to try and even out. Where is the concentration of water higher? Okay. So here, where is the water more pure? Hopefully you can guess it's here. And where is it less pure? In the egg. Okay. And remember, water is going to move from more pure to less pure, or high concentration of water to low concentration. So you can imagine what's going to happen to this egg. And then the opposite, the water is higher concentration of water here, less concentration of water here. So water is going to move high to low out of the egg. Okay, so let's see what happens. She does all the calculations. Okay, and she shows you all that, which is kind of cool. And then what she does is I'm not going to show you that. She shows, then she does it again by switching the eggs. But you only have to do, I'll go ahead, hypotonic solution. And then this one is going to be in the hypertonic solution. She does it again by switching the eggs, but you only have to give the information from her first run of this experiment, and then you're going to answer the question, what happened to the egg that was in hypotonic? You can talk about which way the water went, what happened to the hypertonic, okay? Why? Because the water was moving from what to what, okay? And then here I want you to just show me an arrow. Is the water going in the egg or out of the egg? Same thing, is the water going in the egg or out of the egg? And then last thing, just give me a definition for osmosis, okay? And all the definitions are right here at the very beginning, okay? Diffusion, okay, here's diffusion, and here's osmosis. All right, so I hope you, unfortunately you won't enjoy it as much because you can't do it yourself unless you'd like to try this at home, which is kind of cool. But otherwise, let me know if you have any other questions, and have a good evening.